Good morning. My name is Sri Devi. I am from Ernakulam. I completed graduation and post-graduation in biotechnology and I worked as a biotechnologist. And till 2017, I worked as a medical coder in few companies. In 2017, I started my career as a full-time home tutor. In 2022-23, I worked as a medical coding tutor in a private firm. After that, again more than 1 year, I worked as a private home tutor. In 2025, I joined in Angle BLearn as a full-time home tutor. Okay, that's about my profile. So today our topic is enzymes. So enzymes are one of the finest topic because it's very important topic in case of chemistry and biology. So in this chapter, normally we getting an idea about enzymes and how they act and what are they doing really. Okay, so I think you heard about enzymes in case while we studying in digestive system, also while we studying that liver and kidney. In that while we studying in that or about that organs, we normally learning about some enzymes. Okay, so what are actually enzymes? Enzymes are proteinaceous substance. Okay, enzymes are proteinaceous substance. You got it. What is proteinaceous substance? The substances which are made of protein. Okay, and what are they doing? They are catalyzing chemical reactions of biological origin. That means the chemical reactions which happening inside a cell, tissue or organ. These biological reactions are catalyzed by enzymes. Okay, without undergoing any change. Normally what happens in a chemical reaction? The reactants will change into products. Sometimes the products, from the products we cannot get the reactants clearly, like this. A is a reactant, B is a reactant. Normally a product is made after the reaction, but sometimes it's reversible, sometimes it's irreversible. Okay, but this is not in case of enzymes. Enzymes cannot undergo any change. They remain intact or they cannot undergo any structural changes. Okay, that you will understand in the coming points. So we talked that enzymes catalyzing the chemical reactions of biological origin. That's why they are called biocatalyst. Biocatalyst. What is catalyst? Catalyst is the chemicals using for what? To increase the rate of reaction. Okay, while we consider a reaction, suppose this is x and y axis, the rate of reactions we can represent like this. For initially there is a rise of reaction rate. At a point the product and the reactants. The reactants is equal to product. At that stage a steady state will happen. It will be a sleeping line like this. Then at a point a decline phase will there like this. At this point what? The product will be at high concentration and the reactant at low concentration. So if there is no reactants, the products will not forming. So in that situation, if we add a catalyst to this reaction, again the reaction can accelerate it, or the rate of reaction will increase if we add a catalyst. Okay, so catalysts are the chemicals adding to a reaction to increase the rate of reaction. So biocatalyst means they are the biological catalysts increasing a chemical reaction of biological origin. Okay, you got it. So normally here I use two names of enzymes, amylase and pepsin. Amylase, amyl, that word amyl, which means starch, that means they break down starch. And pepsin, pep means what? Protein. Okay, protein. So pepsin, what they do? They digest proteins. Okay, break down and digest, both are same. That means long molecule chains are breaking down, and if enzymes act there, they break down into small molecules or small parts, like this. This is what happens in the breakdown or digestion. You got it? Okay, I will erase it, otherwise I should not get some space here. Okay. Okay. They are essential for various biological processes in our body. We already discussed that these are reactions happening inside our body, inside the cell. So they are very necessary for the biological process. That point you can relate. Okay. And what are the biological process normally? That is metabolic process. Normally they are metabolic process. What is metabolism? Metabolism we already studying in our lower grade classes. What is metabolism? Normally our body have two type of process. Generally it called as what? Metabolism. What are they? Anabolism and catabolism. Okay, what is anabolism? Small molecules forming into big molecule, that is anabolism. Okay, small molecules join together to form a big molecule, like this. What is catabolism? The large molecule break down into small molecule, like. Okay, for both process we need enzymes. For the aggregation and for the disaggregation they need what? Enzymes. So anabolism we can say that formation of glycogen. Glycogen. What is glycogen? What is glycogen? It's a storage form of glucose. When suppose this is glucose molecules, these glucose molecules aggregate to form a glycogen. This is the storage form. When our body get more glucose, our body store that glucose in the form of glycogen in liver. And while we starving or while we doing any fasting, these liver stored glycogen break down into small molecules, and these small molecules are going to our blood, and it is utilized by the body for energy. This is what happens in our body in daily basis. Okay, this is what happening, and we call it as metabolism. If the metabolism is not proper, the body will show some symptoms that are called lifestyle diseases. Diabetics, blood pressure, fatty liver, these are the side effects of lowered metabolism. Okay, obesity is one of the best example for lowered metabolism. Okay, and these enzymes are very specific. Okay, enzymes are very specific molecules. That means they have particular substrates to react with, and they converting them into what? Into products. Okay, into product. The enzymes react with substrates and they produce some products. Actually, this process is very important for metabolism. Okay, next we are going to talk about the characteristics. Enzymes are very specific substances that you should remember every time. Enzymes are not acting generally, they are very specific. You got that point? Enzymes are very specific. First one is efficiency. Enzymes are highly efficient biological catalyst. They accelerating the chemical reaction. We already talked what is acceleration. That means the chemicals which increasing the rate of reaction. So these enzymes accelerate the chemical reaction. How they accelerating the chemical reaction? By lowering the activation energy. What is activation energy? That is minimum required energy to start a chemical reaction. Okay, whatever type of chemical reaction, that chemical reaction required energy. There is optimum level of energy that reaction required. Otherwise, that reaction will not happen, will not take place. Okay, so that optimum level or the required amount of energy to start a chemical reaction is called activation energy. It's called activation energy. And this activation energy is lowered by the enzyme. Okay, and enabling it to proceed faster even without the enzyme. If the enzyme is not there, the reaction will continue. That is the efficiency of enzyme. Okay, next is specificity. Specificity that I already discussed. That enzymes act on specific substrates. Okay, for example, lipase. Here you can see a suffix ase. In the previous example also you can see ase. amylase, ACE. So ACE, that's a suffix using in case of enzymes. If the ACE is there, that means it's an enzyme. Okay, so lipase, lip means something related to lipid. Lipid means fat. Okay, so lipase act on lipids. Same like lactase. Lactase act on lactogen molecule. Fructose. Fructose, there is a fructase. Fructase act on fructose molecules. So from the word, from the enzyme name, we can understand what is that particular enzyme doing. Okay, these are essential for biological process, and they accelerating the chemical reaction which is involved in the metabolic process. You got it? In this point, you just understand only one thing, that enzymes have specific substrates. Okay, you got it? Okay, enzymes interact with specific molecules known as substrates that we already talked. Again, I am repeating that enzymes interact with specific molecules called what? Substrates, and converting that molecules into products. This is the fundamental basic process happening in an enzyme substrate complex, and that enzyme substrate complex produced is again utilizing for the enzyme product complex. Okay, that you will see in the coming picture. Then you will understand that point, why I mentioned that point here. Okay, this process is fundamental for sustaining life. Why I am repeating this point? Because enzymes are happening, means that enzymes are present only in case of living cells. Okay, so the enzymes are very crucial for all biological process, especially for digestion and excretion process. Enzymes are very important. Okay, next is temperature and pH sensitivity. Temperature and pH sensitivity. Enzymes activity is highly sensitive for temperature and pH. If the temperature is high, the enzyme activity will be high. If the temperature is slow, the enzyme activity will be low. Same like that, optimum pH. For example, pepsin need 1.5 to 2.5 range of pH. So every enzymes have particular pH for their action, for their chemical reaction. Okay, so temperature and pH sensitivity is very important in case of enzymes. And reusable, that point also we discussed in the introduction. Okay, here I mentioned that they without themselves undergoing any change, there is no change, structural changes happening. The same thing repeated here, reusability. That means enzyme not consumed during a reaction, so they can reused for multiple times, multiple catalytic cycles. Okay, this is the basic characteristics of enzymes: efficiency, specificity, temperature and pH sensitivity, reusability. These are the basic characters. Okay, and the mode of action or the mechanism of action of enzyme are of two type. Means normally lot of researches happened in case of the mechanism of action of enzymes, and they put forward two models. So first model is lock and key model. First model is lock and key model. Second model is induced fit model. Okay, here I attached a picture so you can understand it. This is lock and key model. Look at this picture. This is the active site. You can see the active sites. See the structure, and look at the substrate. The structure of substrate almost similar. So they will come and fit at this active site. It forms enzyme substrate complex. Then it will change into enzyme product complex. And after the reaction, the enzymes, from the enzymes the product will release and the enzyme remain intact, or it will not lose their shape. Okay, and they are ready. The enzyme is ready for next catalytic cycles. So this is about lock and key model. In this model you can see that the enzyme is acting like a lock. This is acting like a lock, and the substrate acting like a key. They come and perfectly fit into the lock, so that lock will open. So same like that the process will continue. And here what they suggesting that there is the substrates are peculiar for active sites. Means every substrates not acting on every enzymes. There is particular substrates for particular active sites. The substrates comes and bind with the enzyme, not the enzyme going and bind with the substrates. The substrates coming and binding to the active site. That means here there is inducing a change in the enzyme only because of the substrate. Okay, but this theory was not using nowadays, because the induced fit model was proposed after that. In induced fit model, what happens? In this induced fit model, look at the substrate, look at this active site. What is this? Is the structures are same? No. But look at the second picture. The enzyme adjusts its shape so that the substrate come and bind, and they will react to form a product. Enzyme product complex is formed, and after the reaction the product will release from the enzyme, and the enzyme remain intact. They will ready for the next cycles. So here what happened, that according to the substrate, the enzyme's active site or the enzyme is adjusting itself, adjusting itself for making best products. So this theory is more accepted for the enzyme substrate reaction, because here conformational changes happening to the active site according to the substrate shape. And this is more preferable when comparing to the lock and key model. Okay, that's why it's more accepted nowadays. And more stable and effective enzyme substrate complex are forming in the induced fit model. You got it? More stable, effective enzyme substrate complex are forming in the induced fit model. That's why this model is accepted more widely than the lock and key model. Lock and key model, the active site is more rigid, but in the induced fit model the active site is not rigid, it is adjusting according to the substrate and produce more stable products. That's why induced fit model is more accepted. Okay, and these active sites are otherwise called pockets or crevices, okay, to which the substrates come and bind and produce enzyme substrate complex, that you know, and we can write that procedure like this: enzyme adding to means substrate, enzyme adding to the substrate, then enzyme substrate complex is forming, then enzyme product complex is forming, then the enzyme, from the enzyme the product will release, and the enzyme is ready for next set of cycles. And here the active site is transient state structure. What is transient state structure? That means the active site is not active site, actually the shape of active site is not permanent. It will alter according to the substrates. That is why they used the term transient state structure. The active site is transient state structure because they will change according to the substrate. You got it? You understood both models, lock and key model and induced fit model. Do you have any doubts? You please ask. Both are same, but lock and key model, it's like a rigid active site, means they have a rigid active site in case of enzyme. But the induced fit model, in induced fit model, this substrate, according to the substrate, the enzyme active site will change and they gel very neatly and produce good products, and the products will be more stable, more effective. Okay, that's why induced fit model is more accepted. And in both models, after the enzyme product complex, the enzyme will remain intact, the products will release from the active site, and the enzyme will do the next round of catalytic reactions. Okay, next is the differences. Just I am adding these points for your knowledge, so you can understand the things properly, because it is one of the important topic in case of immunology, biochemistry, microbiology, enzymology. These enzymes are one of the important topic. That's why I'm adding some points here. Okay. So what are the key differences? Rigidity versus flexibility. The lock and key model assumes rigid enzymes. Okay, I already mentioned that rigid active site is there. So the product is produced here may not be long lasting. But that is not in case of induced fit model, which is flexible enzyme active sites are there, and the shape will change according to the substrate. So more stable products are producing in induced fit model. And complementarity. Here in lock and key model, the substrate and the active sites are pre-complementary. That means already that they will match each other. Only that kind of substrates come and bind. So that's one of the drawback of lock and key model, but that is not in case of induced fit model. Here the active site have conformational changes according to the substrate. And next is dynamic. What is dynamics? Dynamics means the induced fit model is more dynamic and accurate representation of enzyme substrate complex formation, because this picture you can understand that things. This is a rigid active site, and here you can see a flexible active site. So according to that, the induced fit model is more accepted, because the enzymes active sites do a conformational change according to the substrate. So they produce good enzyme substrate complex and good enzyme product complex. So the product will be more stable, and the enzyme is ready for next round of catalytic cycles. Okay, these are the differences. Next is we have some kind of some classifications. That for your knowledge I am adding this classification, because you should understand there is no 10 enzymes are there, 12 enzymes are there, not like that. Lots of enzymes are there in our body for doing different chemical reactions. Okay, especially these enzymes are utilizing in cloning, DNA RNA cloning, and genetic engineering, gene mapping. In a variety of fields, these enzymes are very much used product or very much used chemical. That's why nowadays enzymes have more importance in genetical engineering. Okay, oxidoreductases. You see the word ACE, reductases. Okay, oxidoreductase, that means they do oxidation reduction. Kanda, oxidoreductase, that is what they catalyze the oxidation reduction. The example is catalase and transferases. The second type is transferases. What they do? They transfer functional groups. There is functional groups for every chemical. There is a functional group. That functional group is transferred by this transferases. Hexokinases are example. Then hydrolases. Hydro means water. So break bonds via hydrolysis. They do breaking the bonds, hydrogen bonds. Hydrolysis. What are the example? Maltase, lyases. Fourth classification. what they do? Non-hydrolytically acting enzymes. Non-hydrolytically active enzymes. That means they not doing hydrolysis. They doing some other mechanism for breaking down the bonds. They are called, the example are decarboxylases and isomerases. Isomerase. That means isomerism, that you will study in 11th and 12th. What is isomerism? The mirror images of molecules, that is called isomers. So rearrangement of molecules. For example, phosphoglucomutase. Okay, these are this enzyme is example for isomerase group of enzymes. Last group is ligase. Ligase means always coming for joining. Because ligases normally using in cloning for joining the DNA fragments. So join molecules using ATP. ATP I already mentioned while we studied mitochondria. ATP, adenosine triphosphate. Okay, joining some molecules using ATP. So ligases always like biological glue. Why we using glue? To stick the broken edges. So ligase same like that. Ligase, suppose this is a DNA strand and this is a DNA strand. If we give ligase enzyme, it will join like this. The ligase will join this area like a glue and making a long strand. Okay, so ligase are using for joining, and this joining is using the energy of ATP. DNA ligase, I talked about this enzyme, DNA ligase. Okay, these are the classifications of 6 classifications of enzymes. These are the major classifications. Okay, just I am saying these classifications just to understand what all kind of enzymes actually we have. Okay, that's about the enzymes.